Hello. Today we have the pleasure to have two speakers to talk about global health and some of the developments of drugs for malaria and some other neglected diseases. And so Dr. Fabian Gusovsky will talk first about the program of global health in Eisai, which is Japanese pharmaceutical companies that have lots of activities in the US. We've been collaborating for 25 years, maybe. Well, I remember it was early in 2000 when Bernardo was my student. And since then we were working on these drugs for malaria, but he has lots of projects. In Brazil, he had some projects in Universidade of Ouro Preto for drugs for Chagas disease, and also in other countries in South America. He's an Argentinian, so he has the option of to talk in Spanish, Portuguese or English, so you choose. So it's really a great pleasure. He has been visiting Belo several times, and so it has this very important program to contribute to the development of drugs for neglected disease. That's very important. And also after him, we're going to have Dr. Kip Guy, who is the head of the dean. He's the dean of pharmaceutical school at University of Minnesota. And he's currently working on development of a new drug for malaria, Vivex, and they are planning to start part of the study in Brazil. But he has done many contributions on vaccines and drug development for infectious disease. So we're really very happy to have both of you here, and I'm sure it's going to be a very stimulating talks.
Ok. Obrigado, Ricardo, pela introdução, mas infelizmente vou falar em inglês, principalmente porque não tenho tanta familiaridade com a terminologia técnica desses projetos. Mas as perguntas podem ser em português, pode falar em português. Então, we're going to talk about our global health program at Eisai. For those of you who may not be familiar, Eisai is a Japanese pharmaceutical company. It's one of the largest Japanese pharmaceutical companies. It's mainly known these days because recently we got approval for a drug for a monoclonal antibody to treat Alzheimer's disease. It's an antibody that binds to beta amyloid, and that has some help in slowing the progression of Alzheimer's disease. Also, is very active in oncology with several drugs in that space. And finally, our CEO in Japan, he always says that we work in 3 therapeutic areas: neurology, oncology, and global health. So global health is one of the areas of interest for Eisai as a corporation. So we are very, very happy to have the support of the company leadership. So a little bit about what why Eisai is involved in global health. There is a concept called human health care. Eisai aims to realize social good with efficiency, and we define social good as to, these are a lot of Japanese concepts, but sometimes look a little bit strange, but overall they are very very useful to understand why ASI is interested in doing global health, and also social good with efficiency. And this started several years ago. There was a meeting in 2012 of several CEOs of drug company CEOs with Bill Gates, and at that time they committed to form a public-private partnership to combat neglected tropical diseases. This was ratified in 2022. This is Mr. Haruo Naito, who is the CEO of ASI, very proudly showing that we are one of the sponsors of this declaration. In terms of what we do, back in 2010, the head of the WHO was a lady named Margaret Chen. She approached ASI because they were in need of the supply of medicine that is used for the treatment of lymphatic filariasis, filariose, as you say in Portuguese. And ASI very quickly committed to manufacture this and produce it in large amounts, and also distribute it to zero cost to the countries in need. So this drug is called DEC, or diethyl carbamazine, and we received this World Health Organization pre-qualification in 2013, and since that happened, over 2 billion tablets were distributed for free to 29 countries. Interestingly, there is filariose in Brazil. It's about to be eradicated, but Brazil was one of the countries that did not accept ASI's donation because I think it's manufactured by Fabian in Brazil. So Brazil did not need our donation, but many other countries did and still do, and we still distribute that. Okay. One of the things that really helped the development of the Global Health Program was the creation of a fund called the Global Health Innovative Technology Fund, which is a Japanese organization that makes grants for research groups that are proposing developing drugs or vaccines or diagnostics targeting neglected diseases or malaria or tuberculosis. And since this fund was created in 2013, the cumulative funding of this has been, this is a lot of money, so it's probably around $300 million that have been already distributed in various grants. And ASI participates in two ways. One of the ways ASI participates is as a donor of funding to create this cumulative funding. But most importantly, what is most important to us is that we have been very successful in writing grants and securing funding for global health projects that we engage with external partners. And the reason this is important is that we don't want to use ASI's R&D budget for this, because in the drug industry, as you may or may not know, every time there is some sort of financial difficulty, then there is cuts and there is elimination of programs, and we thought that being receiving R&D budget from ASI for that would be very risky. Instead, almost all of our funding for global health comes from partnerships that we engage with scientists all over the world, and securing grants from external organizations. And among those is this GHIT fund that I described earlier. well, now it's no Melinda anymore, but the Gates Foundation, but also other organizations such as the United States Department of Defense and the MRC, which is the Medical Research Council in the UK. So this is the funding part, and in terms of scientific and drug development part, we have a very significant number of partners that go from academic groups. We have a partner with groups in the US, in the UK, in Argentina. We are currently partnering with several in Argentina, in Brazil as well. And we also partner with other drug companies such as GSK and others. There is a number of organizations that are called PDPs, or product development partnerships, such as DNDi and MMV, that are very interesting to provide resources and knowledge about particular diseases, and they are always part of our partnerships. This is a current summary of all the projects that we have in our pipeline. As you can see, our drug DEC is approved and is distributed. We have a drug that is very close to approval for a disease called mycetoma. This is a fungal skin disease that is mainly found in Sudan, in Africa, but in other African countries as well, and as well in India. Is a very disfiguring type of disease. People that contract these infections often have to suffer amputations or disfigurement. We found that one of our antifungal compounds is effective in doing that. We did a clinical trial in Sudan, and we hope to get approval for this drug in the short term. SJ733 is our partnership with Kip that we have held for 15 years, close to 15 years, and hopefully you'll hear from Kip about this project. 1018 is another anti-malarial that I will talk a little bit after this introduction. But we have, again, products against leishmaniasis, filariasis, again, etc., of other, a number of other diseases, and with collaborators all over the world. So, I'm going to provide a short summary of our, probably our most advanced project is KIPS, SJ733. This is probably the second one. It has gone into phase 1 studies now in the clinic, but I'll give you a summary of what this compound is all about. Malaria, probably I don't need to tell you more about malaria. Being in Brazil, knowing how devastating it is, with still lots of hundreds of thousands of deaths every year, and the deaths being children under the age of 5. The parasites belong to the genus Plasmodium. Falciparum is mostly the culprit in Africa, whereas vivax, as we know, in Brazil and the rest of Latin America. And the reason we need novel treatments for malaria is that the existing treatments that are very effective, they induce resistance, and we continuously need to find other avenues to kill the parasite, because the parasite is very smart in getting around the mechanisms that we use to kill them. We also need shorter regimens, and the parasite has multiple stages, and we would like to address not just the blood stage, but all of the stages of its life cycle. Okay. This is an approach that involved a collaboration with the Broad Institute, and we started with a very diverse chemical library that they have, that they call it diversity-oriented synthesis, in which molecules were sort of reminiscing of natural products, but not only that, in each case they synthesize not only they synthesize all of the isomeric forms of those particular compounds. That means that after a single screening round, you could probably identify precise pharmacophores on its targets of action. And what you would like to see in this type of screening is compounds that are very active against not only the native forms of the malaria parasite, but also against forms of the malaria parasite that have become resistant to other types of treatments, such as resistance to other described targets that are already known. So the type of screening that we utilize involved initially high throughput screen that involves a very large number of compounds, retesting in a 4-point dose response curve, enlarging that dose response curve now involving resistant strains to avoid repeating existent knowledge on target identification, and then of course showing that the host or the mammalian cells are not effective at these type of drugs. So using this type of succession of chemical or biochemical assays, a compound emerged early on that we did not know the mechanism of action. This is 1095. And what we did, we established with this compound cultures of malaria that became resistant to this type of activity. So once we generate resistant forms, you do a full genome sequencing of all the genes in that malaria parasite and try to pinpoint where are the differences in the resistant strain versus the native strain. And that, if you are successful, then you probably are very close to identifying the mechanism of action. And what this work resulted is in 3 different resistant lines, and the 3 of them pointed to the same gene. So all the mutations were located in the same gene, and that gene was the phenylalanine tRNA synthase. And then when you compare the activity of a number of analogs that were made from this original compound, when you compare the activity of as antimalarial versus the activity as an inhibitor of this purified enzyme, you see an excellent correlation. Therefore, it is pretty clear that this was the target of this type of drugs. This enzyme is called phenylalanine phenylalanine tRNA synthase, and is involved in the synthesis of the tRNA for phenylalanine. So this is the enzyme, and this is where this compound works, and as a consequence of this inhibition, protein synthesis is impeded and the parasite dies. Some a study by experts in the malaria field did a ranking or evaluation of various novel targets in malaria, and it was observed that this target, FRS, received a very high priority, indicating that it's a good target for novel compounds. And to our knowledge, our compound is the first inhibitor described for this enzyme. as I said, we made several different analogs, and the interesting part in terms of malaria activity is that they are active in all stages of malaria: blood stage, liver stage, and transmission stage. However, in the initial compounds, there were some undesired side effects that with a number of chemical modifications were eliminated. And finally, we came up with a compound called E1018, which has an improved safety profile, and we engaged that into the clinical trials, and that is where we are right now. More recently, what was done is the structural analysis of the interaction of this compound with the target, the phenylalanine tRNA synthase, and indeed it was determined that it binds at the very important pocket, the catalytic pocket, with one portion of the molecule impeding the binding of ATP and another portion binding to a lipophilic site in the core pocket of the enzyme. So in summary, let's just go there. This is a compound with a novel mechanism of action targeting the parasite FRS. It has a low propensity for resistance and is active in multi-stage activity. It seems like there is a chance that we will be able to treat malaria with a single dose as opposed to other drugs that need several doses for elimination of the parasite. In terms of the chemistry, this is something I did not elaborate, but our chemists designed a very cost-effective form of manufacture for this compound. We completed the IND enabling package, and right now we are undergoing phase 1 clinical studies in the United States. This is acknowledgments to all of the members of the team, and I will stop right there. I think we are already a little bit out of time. So what I suggest we do is that why don't we move on to Kip's presentation, and then we could do a combined question and answer session, if that's okay with everybody. Okay?
to U.S. ethics laws, I have to show you this slide. So if anybody makes any money selling this thing I'm going to talk about, I get a little bit of it. Take that as a grain of salt. And somewhere on this large team slide over here, it's actually not somewhere, it's right here in this section, you'll see the collaboration with ASI, but it's not just ASI. The project team, if I take the whole group's about 120 people right now. So I won't go through all of them. I just wanted to point out that it's a large group. And this is, as you know, how we do things in academia, because we don't have Fabian's budget. I'm not going to talk about anything leading up to this. I just did want to say we are targeting the blood stage, and under the current way of thinking about target product profiles, this would be a treating patients drug, not a chemoprotection drug. And as Fabian mentioned, this project's been going on, really since about 1998. This molecule we've been working on since roughly 2008. So in that time frame, the performance parameters for drugs have shifted. And so I'm going to tell you about a drug that actually matches all these pretty well. You'll see how it looks. And we do have a backup now that matches the current version, but it is interesting to see how the target 15 years ago is not the same target now. And so it's good to think about that when you think about drugs in development. And this is largely a blood stage drug. So very similar to what Fabian just said for their tRNA synthetase inhibitor. We did a large screen. I was at St. Jude running translational medicine for them at the time, which is why this compound is an SJ compound. And I've carried it with me now through 2 other institutions in the ensuing 15 years. We looked at about 1 million compounds. We got down to about 1,500 of interest. They represented about 25 series. The one that I'm going to show you in a second actually has progressed into the clinic. We have 2 other series that have come out of this work that are in preclinical that are kind of interesting, but this is the one we've spent the most time and effort working on. So one of the things I wanted to show you, this was actually 2 of the screening hits from the initial screen. And in our shop, we do oral efficacy and oral exposure experiments very early. So almost immediately upon validation of these hits, we looked at them in a mouse model. It's a P. Burgii mouse model, oral dosing of the compounds, 300 milligrams per kilogram, which if you don't work in mice is a whopping high dose. And you can see this compound, which actually was the most potent. It was about a 10 nanomolar cell-active compound, no oral exposure really, and obviously no efficacy, whereas this one had a good bit of exposure. This is time concentration curve in plasma, micromolar peak, about 8 hours of coverage. And it was that relationship, these 2 structurally similar molecules, that had this kind of behavior that made us think maybe we could fix what was wrong with this one, and that's how the project got started. I will point out this would not cure Burgii in a mouse, so we sort of took a little bit of faith that we could do enough work to make it better, and that was a lot of work, which I'm not going to tell you about, because I'm going to try to do this in 15 minutes. So about fast forward 8 years, 400 molecules made in the laboratory, 11 people who worked for me, chemists and biologists working on it, and we got to these 2 molecules. This is 733, which is probably obviously the one that went into the clinic. I want to show you a little bit of the animal data for these 2. So this is now a 20 milligram dose, 20 mg per kg, still about 10 micromolar peak, but now we're getting coverage above the cellular efficacy of these molecules for about a day post-dosing. So all the chemistry work we did significantly improved exposure, and the potency stayed about the same. So these are still kind of 10 to 20 nanomolar potency, depending on what strain you look at. So if we now look in vivo, this is both molecules now in a humanized mouse. So this is a non-skid gaminal cellularly transplanted with human erythrocytes infected with P. falciparum malaria. And we can cure it, take them down to zero at about a 2 milligram per kilogram ED90 dose in the animal. So that got pretty interesting. And more interesting than that, if I look at the parasitemia with relation to time of initiation of therapy, these work within 48 hours of starting dosing. So they're very rapidly acting. That was two of the properties we were really looking for. And then they had a pretty nice safety margin. So going preclinically, non-GLP, about a 40-fold margin above efficacious dosing. So good enough really to commit to that. Did our usual IND enabling package. We had actually no detectable rat toxicology up to a gram per kilogram. So that's, in the US, the maximum dose the FDA will require you to do. And then, stand very close. In the dog, we could detect anemia, again, only at that really top dose, only with 7 days of dosing. And we were able to trace that backwards to forming that hemoglobin. And then when we went and looked really, really carefully in the rat, we could see a tiny little bit of methemoglobin formation that we had missed because it was basically very close to normal. So that was the toxicology we had going into the clinic. Clinical development plan was as follows. We did a phase 1A in the US in healthy volunteers, a phase 1B in Australia, which is a malaria challenge model with falciparum malaria, in healthy volunteers in Queensland. These are college students at University of Queensland for the most part, and they do get compensated for that. And then a phase 2A that was completed in Peru in actual malaria patients in the Iquitos area. So a little bit of that data. When we did the phase 1A, so we got, these are the AUCs in human, in plasma, in the same units I just showed you. And we get pretty nice dose linearity as we dose up until we hit about a 600 milligram total dose in a human. And at that dose, it flattens out. So under our protocol, we did an extra full cohort. It looked like a real plateau. We did 2 sentinel patients at the next dose level up, and then we stopped. That was one of the stopping rules for the trial under dose escalation. But in this single ascending dose study, there were no discontinuations, no serious adverse events. Importantly, no methemoglobin formation, no changes in any hematology parameter. And in fact, we only had 2 drug assignable AEs, and they were both very low level. So for malaria, we're targeting between 1 and 3 doses for clinical use. So we went back and did multiple ascending dose, 3 by 300, 3 by 600. Again, pretty nice AUC linearity, both with total dose and between doses in the 3 sequential daily dose, multiple ascending dose. And again, no real toxicology signal. So during these studies, we figured out it was being metabolized dominantly by cytochrome P453A4. It's the same one that hits a lot of the HIV protease inhibitors. And for that reason, we added a cohort to the trial where we co-dosed patients with cobicistat, which is a 3A4 inhibitor. This is the second generation compound like ritonavir. So Gilead has developed this as a less problematic compound for use in HIV combinations. And again, we got really nice dose linearity, reaching a pretty high total exposure in these patients. Single dose of 600 plus the standard Kobe dose, and no detectable adverse events. So we went from there into the phase 1B. The way this is run, we inoculate these subjects with malaria. We're monitoring them every day for parasitemia. When they start to get PCR detectable parasitemia, we'll let it go a few days up. This is about 10 to the 6th, so subsymptomatic parasitemia, and that's where we initiate treatment with our test drug. If this doesn't work, they get rescued. If it works and then they get ill again, it gets rescued. We never allow them to go symptomatic, basically. They're always treated with standard of care before then. And you can see, as soon as we start treatment, within 24 hours of treatment, we were down to the detection limit for the assay. It does start to rebound on median about 48 hours after therapy. This is basically what artesunate looks like in this model. This is a single dose, non-curative but rapidly acting drug. And we included an expected non-therapeutic dose, and this lets us start building an individualized patient PK/PD model that we can use to direct dosing later on. And that actually has turned out to be important. So we then went into the clinic in Peru. Now we're testing 2 cohorts, SJ alone at 600 milligram dose. That's the projected efficacious dose. And the same dose of SJ together with cobicistat. These are 3 sequential daily doses of 600 milligrams of SJ. And what I'm showing here is parasitemia versus time again. It's a 14-day endpoint, so you can probably obviously see we take rapidly down to 0 detectable parasitemia, no recrudescent or recurrent infection in the 14-day period. We did have 1 subject come up about day 15, and I blew this up for 4 days, so you could see there is actually a little bit of difference between the 2 cohorts. There's no difference in efficacy. It's very rapidly acting in patients, even more rapidly acting than it was in the phase 1b trial. And what we see here is when I pharmacoboost, while the efficacy doesn't change, the speed gets a little bit faster. So the speed of effect is dependent on the Cmax of drug. The Emax is related to the Cmax, but total efficacy is dependent on total exposure, not on Cmax. It's just barely statistically significantly different. So yes, there is a different rate, and I'll show you a different view of this where it's a little more obvious in a second. Again, no real tox. We saw no evidence of ATP4 mutations. This is an ATP4 targeted compound like sapargamin, and no changes in genome really induced by the compound. We were grabbing parasites for genome analysis before treatment initiation and for every recrudescent recurrent case. So short version is that molecule did pretty nicely in terms of the target product profile. It is a rapidly acting short-lived compound, so you need to combine it with something longer-lived in current thinking, and it has essentially no liver activity whatsoever. So looking pretty good for moving forward into the clinic, and we are in, I hope, next week going to get final ethics approval for the phase 2b trial. It's a little complicated because we're using both the FDA and the Peruvian national authority. We have FDA approval to move forward, and we're waiting on the governing IRB for the final approval. But this is what the trial will look like. It's a comparison between a combination of chloroquine to tafenoquine, which is the newer gold standard treatment in the Amazonian basin. Standard of care in Peru is still primaquine, chloroquine, and compared to 3 doses of our compound with tafenoquine, 2 doses or 1 dose. Our PK/PD model actually predicts this 1 dose plus 1 dose of tafenoquine will be curative. So this trial is really powered to determine whether this is comparable to this, setting up for what we hope will be a phase 3 trial for a single dose treatment for P-Vivax malaria. This is all in P-Vivax. And then as we went through FDA, they required us to prove the pharmacological contribution of SJ, so we have to compare to chloroquine alone and to tafenoquine alone. And this is all placebo-controlled, all fully randomized, and set up in order to allow us to design a streamlined phase 3 for approval. We're shooting for April, first patient in across the river in Peru. And I think that's where I ended. Well, I just was going to say, so we're looking for radical cure, uncomplicated P-Vivax, but we have a parenteral formulation for this, a rectal suppository formulation in preclinical, ready to do IND enabling, again in combination with ASI. Both of these have been done together with ASI. We're hoping to look at this for severe P. falciparum malaria, and as it looks like severe malaria for P. vivax malaria is going up in South America, this might potentially be of use in that scenario as well. The compound itself is active in all 5 human infectious malarias, and it's equipotent on vivax and falciparum. And then we're very interested in oral uncomplicated P. falciparum, of course. We're particularly interested in the potential for combining with AL, the current standard of care in Africa. And one of the reasons there are 2 major reasons we're interested in that. One of which is we think we can block the loss of AL by using a triple combination. Dorndorp has suggested this with older drugs, but as safe as ours appears to be, and I didn't mention this, but we are 1 GMP test away from proving preclinical safety in pregnancy, which would mean we can start enrolling pregnant women and women of childbearing potential without strict birth control. And if true, we would be the first registerable drug that could be labeled for use in first trimester of pregnancy. And that's particularly important for falciparum malaria. So those 2 reasons, but additionally, in the laboratory and in the mouse models of malaria, SJ combines synergistically with artesunate, and actually that 2-drug combination will give a sterilizing cure with a single dose. So we think this may actually allow us to reduce the schedule for AL, potentially down to a single dose, but certainly down from the current 6-dose schedule. It's a twice a daily drug given over 3 days, the way that it's used in Africa. So those are the reasons we want to do that, and I'll stop there. I do want to call out, as Fabian mentioned, this has been 15 years of quite close collaboration with ASI. I have found that a joy, and it has been a wonderful working relationship. I hope we will convince ASI to move forward with this. But Fabian and I have done a very good job of raising money through non-corporate sources and Fabian has done a wonderful job of mobilizing resources at ASI in order to get this where it is. And then I think both of us will be happy to take questions.
Said that your drug leads to sterile cure in malaria.
So in animal models, we get a sterilizing cure. In vivax, I was trying to fit into 15 minutes, so I dropped several slides. So we don't affect liver stage in vivax. So what we see, it looks to be clearance of all the blood stage infection, concordant with everything else we've done, and then relapse that comes from quiescent, probably liver stage disease. And we put this data in front of the FDA, so it matches the studies that were done in the 70s for chloroquine alone, pretty closely. So the majority of the relapses start at about 21 days. They run out through about 35 days, and it's about 50% of the patients relapse in that time frame. So it's very much in line with the 70s clinical work. Essentially, the FDA said we don't trust data that old. We want a direct comparison. So they've asked us to prove that that's the case, that we don't affect presumptively liver stage disease. I do think there's still possibility for other quiescent forms of malaria. I'm not convinced it's all hypnozoites, but that's a long side discussion. So we will need a liver stage drug for relapsing malaria for sure. While we have not seen resistance, it's a molecularly targeted therapy. I would not use it in the field outside of a double combination at least, because we will see resistance the way we do with every other targeted therapy. So we're developing it, expecting to take it forward in combination for that reason. I'm actually keep trying to talk Fabian into combining it with their tRNA synthetase inhibitor, because I think that would be very interesting combination. But right now we're doing the kind of conventional thing of older drug, new drug. Anybody else?
I have another question. So, in the
They want to ask you.
Yeah, in the phase 1, you used that inhibitor of CPY, but it seems that in the field trial, you are not using it. So, yeah.
Yeah, so the phase 2A was actually set up to test whether or not we needed the CYP inhibitor, because we preferred the simpler combination. It's cheaper, it's easier to apply, we don't have to worry about drug interactions as much. And because the clearance past 14 days is equivalent, we don't see really any clinical advantage to doing that combination. I think one could make an argument in a severe disease patient that there might be a little bit of advantage to the slightly more rapid clearance. But even in that setting, the clinical clearance is very rapid. It's within 24 hours of therapy initiation, and we're seeing 4 to 6 logs of parasite drop in that time frame. So even in a severely ill patient, it would probably take them into subsymptomatic parasitemia within the first day. So probably don't need it. We can keep it in our back pocket in case, but.
I understand that your trial in Peru, you're in the second phase 2 clinical trial. So did you have the opportunity to look for the asymptomatic patients with very low parasitemia? And how fast you clear the parasite.
So we've not deliberately looked for that, but in both the phase 1B and the phase 2A, we were able to show suppression of gametocytemia and 0 detectable gametocytes, and that's congruent with our. So if you believe that the majority of asymptomatic patients are carrying circulating gametocytes, we probably will clear them. And I don't have enough patients in that bin yet to have any kind of statistical faith in that, but I think it's congruent with all our preclinical data. The compound, what it does to gametocytes is it essentially instantly makes them assume a malformed morphology, and they're not competent either for replication or transmission in non-human systems. And then they get cleared fairly rapidly. I didn't have time to talk about the mechanism, but we believe that rapid clearance is actually driven by host macrophage clearance of infected erythrocytes that's specific to the treated erythrocytes. We have a couple papers out on that, and so it's actually a 2-phase mechanism. If you look at the curves, you'll see a very rapid initial slope that's almost to 0, and then a much slower, about a 4-day total clearance slope. That first phase is what we believe is host immune system driven and spleen driven, and then the second phase is an intrinsic parasite mechanism. And I have a whole other talk on that, but it's kind of a cool mechanism, and we think actually, I would say we believe, we're past think, we're now have a strong hypothesis that this suppresses resistance generation in the field, because the majority of the resistance conferring mutations are not competent for replication. Now, unfortunately, the one mutant that Novartis pulled up for Sapargamin, which is the same target, was one of the 3 that we had on our list that we were worried about. So I think we are going to have issues just like every other drug does with field resistance. So that particular allelic variant, from the genomic data I've seen in the Amazonian basin, is not very prevalent here, whereas it is relatively prevalent in circulating parasites in Africa. So I think basically Novartis had bad luck and we didn't.
question?
I have one for Fabian. I have one for Fabian. In the list of drugs that ISA is developing, you put one for dengue that is still preclinical, and I just would like to know if you have more. You could tell us more about it, and tell you that Brazil is very interested in drugs for dengue, Zika, chikungunya, and so on.
Yeah, so what happened is that this GHIT organization, where we get a lot of our funding, last year opened up. They didn't have antivirals on the areas of interest, and all of a sudden they opened up. And we have two projects, one on dengue, and the other one on chikungunya. And on dengue, they are both very early stage, but what we have done is started screening large libraries and trying to look for potential promising drugs that we don't know their mechanism. It's a very, what we call phenotypic screening, going blind and trying to find novel targets for intervention. So the one in dengue is, we are doing it in Japan. The one in chikungunya actually is a group in Argentina, a virologist in UNSAM, Universidad Nacional de San Martín. His name is Diego Alvarez. He's a very, very good virologist, and he's got a terrific group going. Very early stage. I hope next time I come, I can give you more information.
So I do have a comment, since nobody's going to ask another question. I personally am very interested in the potential of combining this with a vaccine, because I think the co-administration of those in order to deal with latent recurrent disease in vivax might be really interesting. And so I'll just throw that out there for you guys to think about, since that's your space. And
So you're looking more for sporozoite, or and it has to be erythrocytic.
well, yes, I think it would need to be erythrocytically targeted, but I think the challenge, especially in this part of the world, is the 180-day maintenance of effect. And so if you thought about co-administering a blood stage clearance drug together with a vaccine, right, and
Right.
following up the vaccine in your follow-up visits, that might be really good at giving you durable cure. And I think that would be really interesting to think about doing.
Yeah, we here we have a program that we're doing with is to follow up the drop of antibody after the histological cure, after the treatment, and that to make sure how long it, and that would be a good way also to tell whether we're like trying to distinguish asymptomatic from cured people, but so far we don't have a vaccine for erythrocytic stage yet. Any other question? Okay. Thanks a lot. It was really very good, and we hope we can follow up on possible collaborations.